All right, good morning, everyone. Let me switch over. Okay, we are recording this lecture, as always. Today what we're going to do is we're going to start off with some top hat questions. I'll review, and then we'll talk about top hat. And then we're going to get into some of the nitty-gritty today, looking at transport properties through the nephron. What we're going to do is we're going to concentrate on the proximal tubule, the thick ascending limb, and the collecting duct. So this is important today to ask questions. It's very abstract. So let me know if there's anything that you need clarified. It's important that you kind of help me walk along this path, okay? All right. So just a review. Last time we actually talked about the 4 different processes. I'll go back up to this particular slide. We talked about filtration, reabsorption, secretion, and excretion. And with filtration, we talked about the glomerulus, and we revisited the net filtration pressure. So we revisited that particular equation. So let's start off with reabsorption here. Remember, reabsorption is any time molecules, solutes, water, fluid is actually pulled from the inside of the nephron, we're looking at number 3 here, and delivering it back into the blood supply. Okay? That's reabsorption. You're recovering these molecules and bringing them back into the circulatory system. Reabsorption. Question already? Oh, yeah, of course. Thank you. Turning on the projectors around the room. That will help. All right, so reabsorption is recovering solutes and fluids that have entered into the nephron and delivering them back into the body, back into the circulatory system. Secretion is just the opposite. Secretion is actually when you deliver molecules from the blood supply, from the circulatory system, and deliver these molecules, solutes or fluid, into the nephron to be excreted, to be eliminated from the body. So please know the difference between reabsorption, number 3, and secretion. And then the last of the 4 processes is excretion, and hopefully we get to that today. All right. Don't forget that these solutes and fluids, they have to go across these epithelial cells, either across the transcellular pathway or paracellular pathway. And if we're talking about reabsorption, it's being pulled from the tubular lumen, across the epithelial cell monolayer, across the interstitial fluid, and then into the blood supply. Those peritubular capillaries. All right, so what we're going to do is we are actually going to start with the top hat question now that you, couple of brain teasers. I'll start off pretty easy. Let's go with this one first. Okay, what happens to the flow, the glomerular filtration rate, when you dilate the afferent arterioles? So we actually covered this last Friday. Some of you, if you weren't here on Friday, talk to your neighbors. What happens when you dilate that afferent arteriole? What happens to the flow? The flow is called glomerular filtration rate, and it is a flow across the glomerulus. Bullet A, GFR will increase, B, decrease, C, stay the same, or D, none of the above? All right, we're up to 74 students, 75. I'll give you about 10 more seconds. All right. Go ahead and close these. Anybody need more time? Okay. These first ones are always difficult. I'm not liking the duo. You getting on the internet and having to have your phone. How many times I've forgotten my phone? It's really... You good? All right. So go ahead and close this one, and the correct response is A. All right. Good job. 63% of you all got that right. I'm sorry, 78%. 63 students got that right. So good job, good job. All right, so let me show you what slide that pertains to. If you remember this set of slides, what happens if you constrict the afferent arterioles? What happens is that hydrostatic pressure in the glomerular capillary falls and the flow decreases. Remember, GFR is actually flow in mils per minute. All right, so if you constrict the efferent arterioles on the back end, everything upstream is actually going to increase. The hydrostatic pressure in the glomerular capillaries is going to increase, and the flow is going to increase as a result. This is kind of like that garden hose physiology. If you kink the hose, everything upstream, that hydrostatic pressure will increase. All right? If you dilate the efferent arterioles, it's going to decrease hydrostatic pressure in the glomerular capillaries and decrease flow. And then this is what the question that I just asked on Tophat pertains to. If you dilate the afferent arterioles, that actually brings more blood into the area, increasing the hydrostatic pressure in the glomerular capillaries and increasing flow. Okay? So, good job on that. I'm going to ask you, I'm going to skip this one. And this is actually more of a brain, not this one. We did this one on Friday. Let's see here. Okay. All right, this one. This is more of a brain teaser. All right, so think about this. This could be a vitamin or a drug. If substance T is actually present in the urine, does this prove that it's filterable at the glomerulus? So think about the 4 different processes and give me your answer. Yes or no? You can talk to your neighbors. All right. Listening around the room, I hear some right answers. All right, we're up to about 70. I'll give you about 10 more seconds. All right, we're up to 82 now. Anyone in this room need a little more time? Peek around these columns here. All right. So, let's see here. Let's see your responses first. No. Okay, so if someone actually said no, why did you say no? What is another explanation? What could be happening? You said no. Anyone? What's that? That's right. Okay, so sometimes, that's exactly right. Sometimes those filtration slits in the glomerulus are too small. And some drugs, vitamins, may not be able to be filtered at the glomerulus. They go on to the efferent arterioles and then are strongly secreted and end up in the urine. Okay? So, not every drug or molecule is actually filtered at the glomerulus. It could be actively secreted. And what that means, remember, it's transported from the blood into the nephron and then excreted. Okay, so good job. The correct answer is no. Oops. No. All right, good job. How about this one? Substance V is not normally present in the urine. Does this prove that it's neither filtered nor secreted? Substance V is not normally present in the urine. What could be happening? Does this prove that it's neither filtered nor secreted? All right, we're already up to 76. Give you about 10 more seconds. Anybody need some more time in this room? Pretty good. All right. Close this one and let's see, what did you have here? Good. All right, so what could be another explanation? What could be happening? That's right. So it could be filtered or secreted and then strongly reabsorbed, so nothing ends up in the urine. Does that make sense? It could be filtered or it could be secreted, but then strongly reabsorbed, so nothing ends up in the urine. A good example is glucose, right? Glucose is filtered at the glomerulus, and then 100% of glucose is reabsorbed in the proximal tubule. Remember, 100% of nutrients are reabsorbed. You should never have any glucose in your urine, right? That's a really good sign of diabetes mellitus. If you have so much blood glucose levels that it overwhelms the proximal tubule, right, it will end up in the urine. And again, a very good sign of diabetes mellitus. All right, good physiology right there. And speaking of proximal tubule, let's go ahead and start with the lecture for today. Okay, so we've talked about reabsorption and secretion. What we're going to do now is actually we're going to go into, see, this is where we left off. These two slides kind of give you the two Tophat questions that we just asked. Think about that when you get it home. We're going to dive into some of the epithelial cells that line the nephron. Okay, we're going to talk about three major areas. The proximal tubule is at the top. The middle one is the thick ascending limb. and the bottom one is the collecting duct. Now, the reason why I want you to know these mechanisms is this actually gives you a real insight on how the kidney works. Okay, so let's start off with the proximal tubule. This looks very complicated, but I'm going to go through it very slowly. And before we get started, I'm actually going to jump over to the document camera so that you can see the bigger picture. All right, so I've got my Sharpie today. All right, so we have our glomerulus. All right, and our glomerular capillaries, right? All right, so right now we're actually talking about the proximal tubule. That's the first part after the glomerulus. This is the long loops of Henle. So what we're talking about is this area here, the proximal tubule. And what we're doing is we're blowing up, a portion of it, okay? And we're actually looking at the epithelial cells that line this area right here. Okay, so remember, we're going in this direction. All right, so from the inside of the tubule to the outside. All right, and again, we're just looking at this area right here in the proximal tubule. We're going to take a look at the epithelial cells that line. This is the inside of the tube. This is the outside of the tube. So, going back here, this is the inside. The blue right here is the inside of the nephron of the proximal tubule. This red side is the outside, and the blood supply is over here. Okay? So, what you'll notice right away is on the apical membrane, that's the side that faces the lumen, you can see the sodium glucose transporters. These are secondary active transporters that are using the sodium gradient to pull glucose into the cell. Now, you could also use this transporter. This could be the sodium amino acid co-transporter. Same idea, sodium using the sodium gradient to pull amino acids back into the cell. So again, I just want to review from last time. If you only thought about one thing with the proximal tubule, it's reabsorption. Most of the reabsorption happens in the proximal tubule, so it makes sense that, again, 100% of glucose and amino acids are reabsorbed in the proximal tubule. 85% of bicarbonate and 70% of sodium and potassium. So this is all about reabsorption. Glucose is brought into the cell and then it's removed from the cell on the basolateral side using this facilitated diffusion transporter. So glucose is just going down its concentration gradient and then brought back into the blood. We're recovering our glucose and all of our amino acids. So that's how glucose is reabsorbed. All right, yes?
Would you mind giving those percentages again, like 80%?
Yep, absolutely. 100% of your nutrients are reabsorbed. And when I say nutrients, I mean glucose and amino acids. 85% of bicarbonate is reabsorbed, and I'll talk about that in just a minute. Bicarbonate is your most important buffer in your blood. So it is reabsorbed. And then 70% of sodium and potassium, some of your major electrolytes, are also reabsorbed. All right, so let's talk about bicarbonate here. You can actually see on the apical side as well, the sodium proton exchanger. All right, but we haven't really talked about this equation yet. You're going to see this from here on out over and over and over again. This is actually a really important equation, reaction, I should say. Reaction in the proximal tubule, in the thick ascending limb, in the collecting duct, in red blood cells, in parietal cells of your stomach, right? This is also, believe it or not, a very important reaction that occurs in oceans when we think about ocean acidification. All right, so let me go through this fairly quickly. Go back over. Here's the reaction. Carbon dioxide and water react. There is an enzyme that's important here called carbonic anhydrase. Carbonic anhydrase. And it facilitates this reaction. Carbon dioxide and water is converted to carbonic acid. Okay, this is actually carbonic acid. And then that actually is converted fairly quickly to protons and bicarbonate. And these are bicarbonate ions. Now, carbon dioxide is a molecule, a gas, that can freely enter into the epithelial cells of the proximal tubule. Just diffuses, passive diffusion, right into the cells. It reacts with water with the help of carbonic anhydrase and is converted to carbonic acid, which is then converted to protons, these proton ions, hydrogen ions, and bicarbonate ions. Okay, so going back, you're going to see that reaction a lot in many different cell types. Carbon dioxide freely enters into the cell, reacts with water, is converted to carbonic acid, and get this, for every carbon dioxide that enters into the cell, one proton is secreted and one bicarbonate ion is reabsorbed. That's really important. You actually are secreting, you're getting rid of that extra acid in your body. You produce an enormous amount of acid on a daily basis. This is one of the ways you get rid of that acid. Acid is secreted. It's transported across the apical membrane by the sodium proton exchanger. And then bicarbonate is reabsorbed. It's transported across the basolateral membrane by the sodium bicarbonate co-transporter. Okay? Pretty good, right? For every carbon dioxide molecule, one proton is secreted and one bicarbonate is reabsorbed. All right, so sodium is also transported across the monolayer here. It comes in through the sodium glucose co-transporter and through, right, also with the exchanger here, sodium enters into the cell and then it's forced out of the cell by the pump. We all know the pump, the sodium potassium ATPase. Okay, so it uses ATP to pump sodium out of the cell. It's interesting that most of your cells, the pump is actually always on the basolateral side. Always on the basolateral side. Sometimes it's used as a marker just to identify the basolateral side. So the pump is always on the basolateral side. It pumps sodium out. Now, this transcellular movement of sodium, reabsorption out of the nephron, sets up a voltage gradient for the movement of chloride to follow. Right? Chloride's going to follow that sodium movement across the paracellular pathway. So you're reabsorbing salts, sodium and chloride. Which way is the water going to follow? Yep, it's going to go with the solute concentration. Remember, we learned that too. Water's always going to follow the solute movement if there's a pathway for water. So if there's aquaporin channels there, then yes, water is going to follow the salt movement. Pretty good? It's kind of nice to go through this. You all have the tools to understand all of the transporters, these epithelial cell properties, right? Driving forces. All right, so potassium also enters into the cell by the pump, and it's actually removed on both sides, potassium, and across the apical side and the basolateral side. Okay? This potassium pump right here makes sure that potassium isn't rate limiting. It's actually recycling here, right? You need potassium to make sure the pump keeps working, and there's a channel that's right next to it to keep that pump going so that potassium doesn't become rate limiting in this area. All right, so a complicated figure, but walking through it, it's not too bad. There is a video already on your Canvas website if you want to go through this again. Okay. Also, I will post this one as well. All right, so let's skip now to the thick ascending limb. All right, the thick ascending limb. So, going back to our document camera here, we're actually now focusing on the thick ascending limb. It is a little thicker than the rest of the nephron. And again, what we're doing is we're just blowing up this portion. Okay, and we're actually looking at the epithelial cells that line this area. And again, the transport properties are inside to outside. Okay, inside to outside. And we're looking at the thick ascending limb. All right, so glomerulus, proximal tubule, loop of Henle, and now we're here at the thick ascending limb. All right, so going back, here's the thick ascending limb of the loop of Henle. The blue is the inside of the nephron. The red is the outside of the nephron that's closest to the blood supply. All right. First thing you notice is the sodium-potassium 2-chloride cotransporter, my favorite cotransporter. It is actually pulling an enormous amount of salts out of the nephron. Remember, this is called the diluting segment. Fluid that's coming up from the loop of Henle has a very, very high concentration, high osmolarity. Okay? You are now pulling an enormous amount of salt out of the nephron and bringing it into the blood supply. All right, do you guys remember Lasix? Lasix is a powerful diuretic, right? Peeing like a racehorse, this is probably due to Lasix, all right? When you give a racehorse Lasix, that is the most powerful diuretic. They pee an enormous amount. It actually helps with tissue swelling, edema, and nosebleeds, okay? Horses. Lasix is Lasix. Okay, so it's not Lasix like the eye, right? It's Lasix. It actually is, the name is actually furosemide or bumetanide. I better write that down. Anyone want to Google that really quick? Lasix, is it Lasix?
It's with an X. Yeah.
It is with an X, okay. So it is Lasix. And the generic name is furosemide or bumetanide. Bumetanide? Maybe an A or an E, I'm not sure. Looks more like a I. Lasix is the same as furosemide and bumetanide. It all is a blocker of that sodium-potassium 2-chloride cotransporter. Okay? All right. Everybody got that? Pretty good? All right, so we're pulling a lot of salt out. Sodium 2 chloride, potassium enter into the cell. It is transported across the basolateral side by different chloride channels, potassium chloride cotransporter, and again, the sodium bicarbonate cotransporter. What you don't have here is the exact same reaction that we talked about before. This is still going on. Carbon dioxide in water is converted to carbonic acid. For every carbon dioxide molecule, 1 proton is secreted and 1 bicarbonate is reabsorbed. That's still happening here. They just took out the reaction. Okay? So that's why you have the sodium proton exchanger still on the apical side and the sodium bicarbonate cotransporter on the basolateral side. All right, same idea here. All these electrolytes are being pulled out, and sodium is actually moving across the membrane, again by the pump. It's being transported across the basolateral membrane by the sodium-potassium ATPase. In this case, the net movement transcellularly is chloride. Chloride is the net movement, has the greatest amount of electrolyte movement across that monolayer. Sets up a voltage gradient for sodium to follow across the paracellular pathway. So again, in this case, you're reabsorbing an enormous amount of electrolytes, sodium, potassium, chloride. But in this case, water can't follow. There's no aquaporin channels. So water stays in the nephron. This is why this is called the diluting segment. Diluting segment. All right, and if we go back to this slide right here, here's what we're talking about, the diluting segment. The fluid that's entering into the thick ascending limb is about, I'd say, 1,000 milliosmoles. But at the top of the thick ascending limb, only 150 milliosmoles. That's quite a diluting effect. You're pulling out all of those salts, but there's low water permeability. That's the main thing to think about when you're thinking about the thick ascending limb. All right, so why is it, this is a very complicated, why is it that Lasix is a very powerful diuretic? All right, so here's the inside of the thick ascending limb or the collecting duct, and here's the outside. All right, I'm going to draw another one here. All right, at the top, if we only have a few 150 milliosmoles, and the outside has a greater concentration, which way will the water be going? Thick ascending limb, right, there's no water permeability, but if we go into the distal tubule or collecting duct, let's just say there is water permeability, which way is the water going to be going? Out, right? The way to think about this is the inside of the nephron has a higher water molecule concentration than the outside. All right? So we have this enormous driving force for the movement of water out, right? All right, Lasix doesn't allow for the reabsorption of those solutes. So what's delivered to the collecting duct is a lot of salt. What happens to the driving force for water? It gets disrupted, right? So it doesn't reabsorb that much water. And all of it stays in the nephron. What you lose to your environment when you urinate is a lot of salts and a lot of water. So again, this is all about these osmotic driving forces. Kidney is all about diffusion and osmosis. That's why we learned about this in the very beginning of the semester. Okay, so not a whole lot of water is reabsorbed with Lasix. Most of it is peed, it's urinated out of the body. Okay? Is that pretty good? Okay, sounds good, sounds good. So that's how Lasix works. Let's go back. That is not an easy concept, all right? So if you have any questions, definitely let me know. Let's move to the collecting duct. Question?
So can water not move out just into, like, outside of the nephron, or is it in, like, the membrane?
Yeah, so it is actually pulled out of the nephron into the interstitial fluid and then into the blood supply.
Okay.
Yeah, so it also kind of goes throughout the interstitial fluid and into the blood as well.
So, but if it's, if there's no, what is it, aquaporin channel?
Yeah, if there's no aquaporin channels, there's no way for water to leave.
But can it get into the interstitial, like the membrane?
No,
It can't even get into
it can't even get into the interstitial fluid compartment. Yeah,
Okay.
good question.
Okay.
It stays in the nephron.
Okay.
That's right, good question. All right, so let's move to the back end here, finish this up. Okay, so now we're actually talking about the back end, the collecting duct. All right, finish this up. Remember, many different nephron deliver, this is the distal tubule. Remember, there's some regulation between the distal tubule and the glomerulus. And then lots of nephron actually deliver their ultrafiltrate into a common collecting duct. Okay, so this is the cortical collecting duct, CCD, and then we're down here in the medullary collecting duct. All right, so what we're talking about here is we're going to blow up this area right here. Look at the epithelial cells that line this particular part of the nephron. We're talking about the collecting duct now. Okay, pretty good so far? All right, so going back, here's the collecting duct. These are sodium reabsorbing cells, also called principal cells. All right, so just like the name suggests, the blue area right here is the inside of the collecting duct. The red area right here is the outside of the collecting duct, and it's closest to the blood supply. Okay? Looks pretty easy. This is sodium reabsorbing cells. This is a special epithelial sodium channel called ENaC. All right, ENaC. ENaC is pretty easy to remember. E, sodium channel. E stands for epithelial. Epithelial sodium channel, ENaC. And this is actually regulated by a hormone called aldosterone. If aldosterone is present in the blood, then the expression of ENaC channels goes way up. If aldosterone is inhibited, then ENaC channel expression goes down. So aldosterone is a hormone that regulates this channel. And we're going to learn more about this on Friday, actually. You're going to have a video that talks about this. All right, so here's the ENaC channel. Sodium enters into the cell by across the ENaC channel, a highly selective sodium channel. Sodium enters into the cell, and then it actually leaves the cell again across the basolateral side, transported across that membrane by the sodium-potassium ATPase. The net movement is usually sodium across the transcellular pathway, which sets up a voltage gradient for chloride to follow across the paracellular pathway. So you're again reabsorbing all of these salts, sodium and chloride. Okay, this looks like a pretty simple diagram here, but let me just talk about aldosterone again. What aldosterone does is it increases the number of channels, those ENaC channels, on the apical membrane. Okay, so get this. The more sodium that's brought into the cell, it increases the turnover rate of the pump, right? More sodium is removed from the cell across the basolateral membrane, but more potassium is actually brought into the cell and secreted. All right? So when you increase aldosterone, you increase sodium chloride reabsorption, but you also increase potassium secretion. Confusing? Questions? Yes, I will say that one more time. All right. With aldosterone, what happens is you increase the number of these ENaC channels right here on the apical membrane. That brings in more sodium into the cell, which increases the turnover rate of the pump. It increases the activity of the pump to remove that extra sodium, but what it does is it also brings in extra potassium into the cell, increasing potassium levels, which are then secreted. They're actually transported across the apical membrane by potassium channels, so you increase the secretion of potassium. So what's cool about this is if you overloaded on potassium, right? Let's just say you ate 50 bananas, or you ate like 3 bags of apricots, which are also high in potassium. What it does is it triggers the release of aldosterone, right? Triggers the release of aldosterone, so you get more sodium chloride reabsorption, but you also get enhanced potassium secretion because you have to get rid of that excess potassium in your blood. Why? Potassium, high potassium extracellularly, can disrupt neurons and, more importantly, could stop your heart, can cause arrhythmias. All right, so your body tightly regulates potassium in your body. Questions? That's good regulation right here. Good regulation. All right, so how are we doing? Oh, it says, okay. Let's actually start on excretion here. Let's just talk about some of these parameters. These are some excrete from your body. This is a process called urination or micturition. All right, micturition. So let me just write that down. Okay. If anybody wants to come up and get some pictures of these two. Urination. Fancy name for urination is micturition. Micturition. All right. All right, so going back to our slides here. All right, so we have some important nitrogenous wastes that I want to make sure that you all know that we excrete on a daily basis. Ammonia is one of them. Urea is another. These are nitrogenous wastes that we get rid of out of our body through the process of micturition. Okay, urea is actually an important osmolyte in the kidney though. We're going to talk about that when we talk about vasopressin later on, but it actually helps to maintain that osmotic gradient that's established by the loop of Henle. We'll talk more about this on Wednesday. So just know that urea is an important osmolyte in the kidney. All right, uric acid. A lot of uric acid is produced and excreted, especially by birds, right? Uric acid is eliminated in the white pasty, it's by birds. Uric acid is actually a breakdown product of purines like guanine. Okay. Allantoin is also an important molecule. Nitrogenous waste, that's eliminated. All right. And trimethylamine oxide. Trimethylamine oxide is a molecule that gives fish its very fishy smell. It's also an important nitrogenous waste. Oops. Okay, and then finally, creatine and creatinine. Now, we talked about creatine before. Creatine is a molecule that is within our muscle, our skeletal muscle cells. Remember creatine and creatine phosphate? Creatine phosphate, there's an enzyme that converts creatine phosphate to creatine, which phosphorylates ADP, right? Phosphorylates ADP, produces ATP so that it buffers any kind of rapid depletion of ATP within the cell. This is the molecule that I said not worth your money when you go to GNC. We can argue about that only because it really is only effective in the first 6 seconds of any very intense workout. All right? Now, here's where I want you to realize creatinine, though, is a very important molecule that is handled in the kidney very specifically. And it is a breakdown product of creatine. Again, this is creatinine. Now, here's what I want you to, the take-home message here. Creatinine is a molecule that is freely filtered, but not secreted or reabsorbed. So I'll say that one more time. Creatinine is freely filtered at the glomerulus, so it enters into the nephron, but it is not secreted or reabsorbed. So everything that's filtered, all of that creatinine that's filtered, ends up in the urine. So you might, if you've worked in the clinics before and you look at kidney function, some of the parameters, the markers used to look at kidney function, creatinine clearance is one of them. If you can measure clearance for creatinine, and we're going to learn about the clearance equation later, basically it's a marker for glomerular filtration rate. Whatever is filtered ends up in the urine. It's very easily tested. All you have to do is test for creatinine in the plasma, test for creatinine in the urine, and then plug it into the equation, and that is giving you GFR, the flow, glomerular filtration rate. This is called creatinine clearance. It's an important marker to measure GFR. All right. So, how are we doing on time? Actually, we are pretty much done. Let's finish up with excretion on Wednesday. There's only 2 more slides. I should be able to go through this very quickly. And Wednesday, we're actually going to be talking about how the loop of Henle establishes that osmotic gradient in the kidney. All right? Have a good Tuesday. I will see you on Wednesday. All right. This off. Okay. Okay. Okay.
I need this one.
Ah, yes.
The drawings or the